Okay, so it's 4 o'clock. So we're going to get started. Biology 107. So good afternoon, everyone. Let's get into this. We're going to basically pick up where we left off. Topic 1, which was an introduction to the cell and the scientific method. So picking up where we left off, we were talking about life. We were talking about what a cell was. And I think I kind of actually meant to finish off on this slide here last day, is just to say, what is cell biology? And cell biology is the study of cells, obviously, but it also involves many other things, cell parts, things that can infect cells, such as viruses. And of course, there's many types of cells. We covered a few last day. We talked about plant cells and animal cells and bacterial cells and things like that. And so all of these things that you can see on this slide are some of the things that we'll be talking about in this class. So different organs, different molecules, and as I mentioned, we'll be talking about viruses as well. So here's a little diagram showing the size of things, and I have something I want to share with you here in a moment. A little tool that's on the internet that's very, very cool. But you can see that they're showing way here on the right side, things that we can see with our eyeball. And most of cell biology actually falls into this category in this box here. So there's a few things we can kind of see with our eyes. You can see there's some very large cells, such as a human egg or a paramecium. And these things are just kind of just within that range of what we can barely make out with our eyeballs. Most of these other things are going to require some sort of microscope or other tool to study. So what I did want to show you was this tool here. It's called the scale of the universe. So what I'm going to do is just switch my screen here and get over to the scale. This is just a really cool thing I like to share every semester. It's called the scale of the universe. It was put together by a couple of guys that are twins, I guess, and they're computer guys. They do a bunch of animations and things like that. And this is one they did. It's called the scale of the universe. And I'll show it to you. It's very, very neat. So here it is. I apologize for the annoying music. Maybe I can turn that off. There we go. And what they did is they decided, hey, let's see how big things are. So you can see, for example, there's a human there, right? You can click on that, gives you a little information. Average human is 1.7 meters tall. A little bit of information. He says, I'm going to assume that you are a human. I'm a human too. Did you know there are over 7 billion of us? And anyway, you can click on the objects so you can get an idea of a little bit of information about them. And you can see, for example, sunflowers can be taller than humans. This thing is cool. This is the world's largest flower. So you can learn a little bit. Anyway, you can zoom out and see how big things are. You can see there's a few different animals, and there's a T-Rex, and this whole thing is just, like I said, it's very cool. I actually downloaded the app for my phone. I think I had to pay $1 for it. And it's just one of those things that every once in a while I'll pull it out and just play with it. You can see an oak tree. If you zoom out really far, you can see some man-made objects, Statue of Liberty, the Titanic, the Eiffel Tower, and it has these circles here, so 10 to the 3 meters, that's 1,000 meters. And there's the Great Pyramid of Giza, Hoover Dam, the Half Dome, and we can zoom right out and see lots of things. So this here, I don't know what that is. I don't even try to say that. It's known as Earth's second moon, so some sort of asteroid thing that orbits the Earth. So there's something that maybe you didn't know about. Anyway, I'm going to just zoom out really quickly, show you a few cool things. You can see there's some other moons. There's Pluto. And of course, Pluto is no longer a planet. It's a dwarf planet. Whenever they bring that up, it kind of bums me out. It just seems like Pluto was, kind of cool, and now it's been demoted. But anyway, there it is. You can see Mars and the Earth. I'll show you a couple other cool things, and then I want to zoom in and talk about the microscopic. I want to show you this. There's Earth, and there's the Minecraft world. So apparently the Minecraft world is actually bigger than our actual planet. Kind of blows you away. But anyway, like I said, you can zoom out and see the different sizes of stars and nebula, and it gives you an idea of the perspective scale of things. I mean, this is just crazy how big some of these things are compared to our own planet. But what I wanted to do was zoom right in and show you a few small things. And like I said, this is, you can just click on the link on the PowerPoint and check some of these things out. But I want to point out a few small things, and we're going to come back and talk about these a little bit later. So where's our human? There he or she is. And we can zoom in. You can see there's a few other things there. And we're going to zoom in and kind of get to the microscopic area. So there is one really large bacterium you can see here that is basically visible with the human eye, which is incredible. Not very big. Like this here's a duckweed. If anyone here has ever had an aquarium duckweed will sometimes take it over. So you may know what that is. So right here, we'll stop, and you can see that there's a human egg, an ovum, and a paramecium. And I mentioned that those were kind of just within the range of what a human eye can make out. And right here is a human hair. So that is kind of a good measurement because a human hair is pretty much the skinniest thing most of our eyes can make out. Some people have a little bit better vision, some people a little bit worse. But that's about the resolution of a human eye, which is about 100 micrometers. And we'll talk about what a micrometer is eventually. This is showing, you can see this gray ball. It's about the same size as that. You can zoom in. You can see all sorts of cool things here. We have chloroplasts, white blood cells. There's a mitochondria, and there's E. coli. Yay, E. coli. I like E. coli. We'll talk a lot about E. coli. Human chromosome, and now we're getting down here. You can see it says the smallest thing visible to an optical microscope. That's kind of a normal light microscope. You can see there's 1 or 2 viruses that are able to be seen without, but they're going to just look like a speck on a microscope. Most viruses are smaller. There's HIV, hepatitis B, and they must be seen using an electron microscope. Anyway, you can check this out on your own. You can zoom right in. You can see there's phospholipids. You can see how big an atom is, and we've got electrons and quarks and all sorts of interesting subatomic particles and whatnot. But anyway, check it out on your own time. It's kind of a fun little thing to see here into science and whatnot. So like I said, right here, 100 micrometers, that's about what we can see with the human eye. And 100 micrometers, by the way, is equal to 0.1 millimeters. Or it's also equal to 0.0001 meters. So very, very tiny, about the width of a human hair. All right, so that's where I meant to be last day. So only 8 minutes behind the semester, not too bad. We're going to talk now about a little bit about the history of the discovery of cells and a few points to make along the way. So the person who probably was the first person to discover cells, there may have been others, but this guy published about it was Robert Hooke. And this is back in, I believe it was the 1600s. I think I have a date there. There's the date, 1600s. And the telescope had been invented sometime that 100 years. And of course, it didn't take long before people started to invent microscopes. At this time, there were probably something like 5 in all of England. And anyway, he was a rich guy, got a hold of one, and had a telescope as well. And so he was able to look at a few things, and he published about them. I see Trevor has his hand raised. I'll get you in a second there, Trevor.
Sorry, I just wanted to say you're still on your browser tab as far as the screen share.
I'm still on my browser tab. Okay, that is good to know. Thank you for telling me that. That was Robert Hooke. Yeah, so there's Robert Hooke, and he had a telescope and a microscope, and he basically just published what he saw, and that is that's kind of part of how people learned about things back then, by people publishing. And like I said, he was relatively rich. He was able to publish, and he saw a few things. He was looking at bugs, so no surprise there. They're kind of cool to look at under a microscope. Snowflakes. And one of the things that he also looked at was cork. So cork is actually an aquatic animal, and it's made of cells. And he looked at them, and he was like, hey, that kind of looks like cells. But when he said cell, he kind of meant like, if you think of a cell in a prison or a monastery. You know, he was familiar with cells where monks lived, where they're in all these rooms were in a row, and so he coined the term cell. So, discoverer of cells, or at least the first person to publish about it that we know of, 1663. So a little bit later, but contemporary with Hooke, was this other guy here, Antony van Leeuwenhoek, another rich guy, and he was Dutch, so he was in the Netherlands, and he had a lot of time on his hands, and he made these microscopes. So these microscopes here are not very sophisticated. If you take a look at this, we have basically, sorry about that, go back. There's a lens right here, and then everything else is just a frame. So this is really just a sophisticated magnifying glass. There's a lens, there's this apparatus here where the specimen can go on, and you can kind of focus it by turning those screws and knobs, but pretty much all you're doing is holding it up to the light and using that lens to look at it. Now, it turns out Van Leeuwenhoek was very, very good at making lenses. He was probably better than anyone else on the planet at the time. In fact, he was better than anyone else at making lenses for 100 years after his death, because he took the secret to his grave, which was kind of interesting. But so what was interesting about Van Leeuwenhoek is he started to look at things, and he looked at what Hooke looked at, and he also started to look at things that were maybe not so obvious. So for example, you can see over here on the left, he looked at rainwater. Now that's the kind of thing that you would just assume, or at least back then, you would assume rainwater. What can be more pure, right? Water is a very pure thing. So I looked at rainwater. I found this is not obviously the sample he looked at, but this is a sample of rainwater that somebody posted on YouTube. And he's like, whoa, what is going on there? There's little something swimming around, right? So the term animalcules came out. So you think they're kind of like little animals, but they're small like molecules. So that word animalcules, kind of cute word anyway. That's what was described. And he did use this in Dutch, so I don't know whether that's an English translation or not. But this was a very surprising thing to find all these creatures. And so he was probably the first person to observe living cells, because Hooke was looking at dead things, right? Dead cells. And Van Leeuwenhoek went along and he saw all sorts of things. In fact, he made a whole bunch of biological discoveries. Think about all the things that are microscopic that we had no idea there were cells there, right? So he looked at blood, and he discovered blood cells. I mean, what an incredible discovery. Humans had no idea these things existed before. Amazing biological discovery. He discovered bacteria, which shows how good his lenses are, because they're very, very small. He also looked at his own fecal matter and saw some weird things in there. And he looked at his fecal matter when he had diarrhea and noticed it was different. He didn't have any connection with disease, because there wasn't a concept of the germ theory at the time. And he looked at sperm. So there was another kind of interesting biological discovery that people didn't know how that worked before. So they're able to figure it out. So at this point, people have discovered cells, and people are wondering, some of them are obviously kind of like animals, but then when you look at cells and you find them in plants and fungi and all these other things, the question is, what are they? What are they doing, and how are they important to biology, right? So one of the theories that was also floating around at the time was this idea of spontaneous generation. And this is an obsolete theory, by the way. But before that, it was this idea that, and not everyone took this seriously, but there was a vigorous debate in the scientific community about this as to whether this was a thing, right? Like how does life begin, right? If you think about, I mentioned sperm and conception, right? People had an understanding that, to make babies, you get a man and woman together, right? And probably had something to do with the, with the whole process there. But how does life begin? And so this was what spontaneous generation was all about, and kind of proposed that if conditions were in the right way, that something could appear. So I'll show you an example here. I have a recipe, and you can find these recipes on the internet. So this is a recipe from the 1600s for making mice. And the recipe is like this. You get a big jar, and you put in some wheat, and a pair of sweaty underwear. I'm not really sure why that's part of the recipe, but apparently it is. And then you store it uncovered for 3 weeks, and then what do you get? You get mice. So I know this is kind of silly, right? And I mean, we, at least, back at that time, many people were farmers, and you had an understanding again that to make a baby mammal, you needed a male mammal and a female mammal, and all that. And if you sat around and waited, you would see the mama mouse come along and give birth to the baby mice, right? But people were debating this because people were not being very scientific at the time, or at least we were just beginning to be scientific. We were observing things carefully and watching and experimenting. And so this was a legitimate idea. For mice, this is pretty easy to disprove, but if you think about all the other biological entities out there, some of them are very, very weird. And I'll show you one in a moment here. So people were debating this, people were getting more scientific, and you can imagine the more scientific people were getting, the more skeptical they were getting about this. So enter this guy, Francesco Redi. He was a scientist, and he was looking at maggots and flies. And so I just want to show you what was going on here, right? So back then there were no fridges, and if you wanted meat, often you went to a butcher shop, and even the cleanest butcher shops had flies and maggots. Obviously the cleaner ones had less, but this was a thing that they were trying to figure out what to do with, and nobody knew where these maggots came from. They just seemed to miraculously appear. So it seemed like a case where you had some actual spontaneous generation going on. So again, here's this case where, according to human knowledge, people did not know what a maggot was. In fact, you may not know what a maggot is. A maggot is a baby fly. And if you look at it, there's the maggot and there's the fly. They don't seem like they're the same species. They don't look anything alike. People had not really investigated carefully the fly life cycle. So he's like, okay, I want to, these things can't be just appearing. There must be something going on. Must have something to do with the flies. So basically what he did is he covered, he put the meat in a jar and then covered the jar up. So you can see in the second picture here, there's the image of the jar. And so you got a stopper there, and of course the skeptics are like, well, of course there's no maggots, there's no fresh air getting in there. And so he just said, okay, fine, I'll cover it up with a cheesecloth instead. So a cloth is going to allow air in there, and the maggots did not end up on the meat. But interestingly, the maggots did end up on the cloth, which led to the hypothesis that the maggots came from fly eggs. And of course, eventually that kind of thing was proven. So what does this have to do with cells? This has to do with all living organisms. And one of the things, of course, about cells is that they're the building blocks of life, and where do they come from? And so like I said, for animals, people were kind of pretty ready to believe that, a mama mouse came along or a mother fly. But what about these really small cells, such as bacteria? Where are they coming from and what are they doing? This is a really important question, right? Bacteria, are they doing something for us? Are they making us sick? These were good questions at the time, and actually these questions went on for a long time, and you can see all this is now skipping forward to the 1800s. So this is more than 150 years later, and now a new historical figure here is Louis Pasteur. So this was a French scientist. And he was a very good scientist and very famous, particularly if you're from France. So he wanted to answer the question, is where did microorganisms come from and were they present in the air? So he came up with this experiment that's now called the famous swan-necked flask experiment. So swan-neck flask, and I'll show you what that looks like in a minute. So you can see what he has there is he has this flask and he has some media in there. So media is basically water and some nutrients. So you can think of it as like a soup for microorganisms. And you can imagine if you had soup and you leave it in your pot and you let it sit out for a few days on the tabletop with no lid, something's going to get in there. It's going to get mold or bacteria and it's going to get putrid. So he came up with this idea, and we know now that why does it get putrid and spoiled is because you have spores and bacteria in the air, and they're gonna spoil it for you. So what he did is he took his flask and he heated up the glass and melted it like this in this swan neck shape they call it. So the whole idea here is that air can get in. Remember, all the skeptics are concerned about air. And so you got to have an open system so the air can get in. And he's boiling it, right? So boiling it to sterilize it. And then you know, we're going to wait and see. Is this going to spoil? Are bacteria going to spontaneously generate and appear, right? And so they didn't. And in fact, if you take a look, there's one of Louis Pasteur's flasks. This is one of his flasks. So this has been sitting in Paris for more than 150 years now, and no bacteria have appeared. The very same flask. Now, what is happening here? Well, you can imagine we've got these little dust particles, right, in the air, and gravity eventually is going to bring them down, and then they're just going to get stuck there. They're going to get stuck in the neck of the flask, and that's exactly what happened. And you can show that there are bacteria in there. You can collect those dust particles, and what you can do is you can break the flask, or you can kind of tip it in such a way that it's going to put some liquid in this neck part, and then let that go back in the flask, and then you're going to have spoilage of the media. So a very famous experiment showed, you know, where cells, that not necessarily where cells came from, but that they don't just appear miraculously. And spontaneous generation is well, it's a disproven scientific theory. So this led to a lot of people studying cells, and we have Schleiden and Schwann were, you know, somewhat contemporary at that same time. Schleiden and Schwann were, one of them was a botanist and one of them was a zoologist. I can't remember which was which. Anyway, they met for lunch, and they were talking, and you know, the one guy said, you know, every plant I've looked at is made of cells. And the other guy said, well, every animal I've looked at is made of cells. So they came up with what we call the cell theory, which is a modern kind of tenet of biology, kind of something that we probably call a scientific law now, meaning that it's been shown to be true for a long, long time, and we don't have any reason to believe it will ever shown to be not true. So this was their tenet, that all living things are made of cells, one or more, that the cell is the basic unit of life, which we kind of talked about last day, and that all cells come from pre-existing cells. So basically that spontaneous generation is just a bunch of junk. Nowadays, people might add a few things to the cell theory, kind of modern additions, you know, about that all cells have hereditary information in the form of DNA, that cells are involved in energy metabolism, and that the cells are made of basically the same things. We got proteins, carbohydrates, nucleic acids, and so on. And we're going to talk about this next day. Okay, so quick break. I told you last day that I want you to download this app, and we're going to do a Kahoot right now. So I have a very short Kahoot, just so that people can try it out and test some of your knowledge. It's a 3-question Kahoot. So load up the app. I'm going to load up Kahoot. Where is it? There it is. And it should give you a number here in a moment. It'll give you a number of what game to type in. So it says, get ready to join, loading the game pin. Usually it doesn't take so long. There it is. So what you need to do to join the game is load the app, or you can go on your web browser if you wish, and you're going to type in that number, and you can join the game.
Sorry, I can't see the number.
Okay. We can't see the screen.
You can't see the number. Is the screen sharing not properly? Okay, let me, can't find Zoom. Let me try that again. Oh, the sharing ended. Okay, try that again. Nope, sorry, wrong one. There it is. Does that work?
Yeah.
Okay, sorry about that. So game pin is 695-4301. All right, I'll give you another 10 seconds to join here. All right, this is an easy one. Hopefully you guys will get it, but like I said, I'll make them a little bit harder as we go along, and give you guys some questions to get to know the information. But this is just introduction. Question 1, who discovered and named cells? Okay, Robert Hooke, good job. One person guessed Louis Pasteur. He was the person who disproved spontaneous generation. Hooke was the one who looked at cork. All right, and so the way Kahoot works is the faster you answer, the more points you get. So if you want a friendly competition, see if you can rack up some points. Number 2, what is your instructor's name? Okay, 100%. So no one guessed Dr. Salmonella or Dr. Microorganism? Come on. Anyway, just being silly here. All right, question 3. True or false? Labs start this week. Okay, so the answer is false. Labs don't start this week. They will start next week. So this Thursday, I will, probably what I will do, the only thing I will do is send you an email just with a little bit of information about the labs, but there will be no assignments this week unless you have not done your plagiarism recognition course. Then that would be your assignment for this week. And I don't know how long that takes. I think somebody told me about an hour and a half or something like that. So if you have not done that, do that. If you have done it, then there's nothing for this Thursday for you. So let's see what the score looks like. This is all just for fun. If I had prizes, I would give them to you. But bronze for Michelle and silver for Tristan. Oh, the suspense. And Levi got gold. And you can see the runner-ups. Okay, so I'll switch back to PowerPoint. So good job, like I said, they're going to get a little bit harder in the Kahoots and hopefully help to assess your knowledge and help me to figure out, whether I need to slow down or speed up or whatever. All right, so today we're going to talk about a little bit about classification of cells and types of cells and whatnot. We are going to get into these things in a lot more detail. We're going to talk a lot this semester about eukaryotic cells and prokaryotic cells. So eukaryotic, by the way, this is Greek. So eu means true, and karyote in Greek means kernel or nucleus. So eukaryotic cell is the big thing on the left. It means true nucleus, and that would include us. We have a nucleus in our cell. Prokaryotic, so karyote still means nucleus, hasn't changed. Pro actually means before. So I'm not exactly sure who named these things, but when they named them, they figured that bacterial cells, prokaryotic cells, were more primitive and maybe predated eukaryotic cells. So that's where the name came from. I don't know what no is in Greek, but I learned these words anyway. So we're going to talk a lot about the differences between these things. At the moment, you can see the two main differences, right? One is that the eukaryotic cells have a nucleus, and two, they're usually a lot bigger. Usually kind of being one of those biological words you could put in front of every sentence in biology. You could say usually this, usually that, because there's always weird exceptions. And that's one thing that makes biology lots of fun. So here's kind of a typical prokaryotic cell. This includes two groups, the bacteria and the archaea. And I will be talking a lot about them as we go on. I think topic 4, we're going to talk about prokaryotic cells in a lot of detail. You can see there's no nucleus, but it does have a lot of features. It has an organelle for swimming called a flagellum. It's got lots of cell surface features. It's got ribosomes. It has DNA. So these are fully functional cells. There's a eukaryotic cell. This is representing an animal cell. You can see it has a nucleus, has a whole bunch of other membrane-bound organelles, and all sorts of other features that we're going to talk about. So as I mentioned, I want to talk a little bit about classification today. And you can see that when we look at cells and organisms, we can classify them in different ways. So one way is that we can say we've got a prokaryotic or a eukaryotic cell. Another way is sometimes we talk about domains. And there are actually 3 domains of life. And I'll show you another image in a moment. And the eukaryotic domain actually includes all these things that you're probably familiar with. So you've got animals and plants and fungi, something called protists and algae. And then, of course, as we talked about last day, there are things called viruses. They don't have cells. They're not usually considered alive, so I kind of put them in their own special table. But they are worth mentioning because biologically we do classify them, and they do impact us in serious ways. So here's our 3 domains of life. And this particular diagram that you're looking at is called a phylogenetic tree. So phylogeny is worth defining. Let me put this in the corner here. Phylogeny. So phylogeny is classifying things, classifying organisms, based on, what's the word I'm looking for? Relatedness, I guess, is a good word. So what does that mean? It means, right, you know, in a modern biological sense, what phylogeny means is how similar DNA sequences are. So how similar genetic sequences, I guess I should have put genetic rather than DNA, so I'll just cross that out. Genetic sequences are. So I just got to apologize for my writing. It is a little bit neater. Usually it's just not as easy for me to write on a computer screen. I'll do my best to see if I can improve that. And but hopefully you can read that. So in this case here, what they've done is they've looked at all sorts of different organisms, and you can compare, like I said, their genomes, and you can see how similar things are. And this, people started doing this back in the 1970s, and they used to just have one group they called bacteria, and they realized that when they started to look at these microorganisms, that one group was quite a bit different from a cellular level and a genetic level. And so they broke these things into 2 groups, these prokaryotes, and we have the bacteria over here on the right and the archaea over there on the left. And like I said, we're going to talk a lot about the archaea in topic 5. But biologically, they're quite a bit different from bacteria. Their phospholipids are different. The way they organize their DNA is different. There's a lot of different things around, about them biologically that make them unique. The only thing they have, you know, the thing that's similar to bacteria, obviously, is no nucleus. And then the third group, of course, are the eukaryotes, which have a nucleus. And you can see up there, I've got the plants and animals and fungi are all highlighted, because they're probably the kingdoms that you're familiar with, but there are quite a few others that fall into other categories. So there's our domains of life: our bacteria, our archaea, the eukaryotes. We've got the animals, the plants, and the fungi. And I also have this group here, the protists, which I'm going to define right now for you. If you don't know what a protist is, so protists. So unfortunately, protists are poorly defined. I'll say this out loud and then I'll write something. But basically, a protist is a eukaryote that is not an animal, plant, or fungi. And that's really what a protist is. So these are eukaryotic organisms that are not plants, animals, spell properly, animals or fungi. I'll say one more thing about them, is that they're usually, but not always, usually unicellular. So sometimes people will put algae into their own group, but a lot of times algae are grouped in with protists. Now, algae are not plants. Plants have all sorts of organs and structures that algae do not, even though they are photosynthetic and closely related to plants. But sometimes people, like I said, include them as their own group, and sometimes people group them in with the protists. So here's another way to classify things by taxonomy. So there's that word taxonomy up there. I'm not going to define taxonomy on the slide here because I don't think I have any room, but taxonomy is classifying things based on physical traits. So I'm going to give you a couple of examples to show you how this works and kind of just highlight what is important for Biology 107. So here's a human, and a human is part of the domain eukarya, because we have nuclei in our cells. We are in the animal kingdom, and animals are multicellular heterotrophs, so we eat things, we're multicellular. I think with animals as well, part of the classification is that we can move around, and there's a few other things. So most of these classification things usually have a big list of things that distinguish animals from fungi, let's say. Because fungi are also multicellular heterotrophs, but I'm just putting a very basic scheme here. We're also chordates, which means we have a central nervous cord. We are mammals. Mammals have hair. We have, our females have mammary glands that feed milk to our young, and those are some of the features of mammals. We are primates. We have opposable thumbs, and we are hominids. We have a large brain case, and we are apes because we have no tail. That means we're different from monkeys. That's the difference between apes and monkeys, by the way. Monkeys have tails and apes do not. So that would include chimpanzees. Homo means human, and you probably know that we are Homo sapiens. So sapiens means thinking or wise or sentient or something like that. We get to name ourselves, right? That's the nice thing about this. We get to call ourselves wise, whether it's true or not. So let's take a look at another organism here, and I'll show you another example. Second example is a dog. So if you take a look, a dog is in some of the same categories as humans. It's a eukaryote, it's an animal, it's a chordata, it's a mammal, it has hair. But that's where we realize it's a little bit different. So dogs are in a different order. They're in order carnivores. They have, and that's based on their teeth structure and obviously their diet. Although dogs, by the way, are not fully carnivores, they do eat a lot of plants. And if you take a look at the ingredients of your dog food, you can see, they do digest a lot of non-meat items. They're in the family of the canines, which apparently means dog-like. That's the best information I could find about what canine is. So it includes foxes and coyotes and dogs and wolves, of course. So the genus for a dog is Canis. That means dog or hound. And the species is Lupus. So you probably know that dogs and wolves are technically the same species. And there's the word lupus. So maybe you've seen, or read those books, the Harry Potter books. And there's that, in the third book, there's Professor Lupin. And the big mystery, or one of the big mysteries around the book is that he's a werewolf, right? And so the name Lupin, right? The author, she used, the word lupus. It means wolf in Latin. And if you knew your Latin at that point, you would have probably known that this guy was some sort of wolf or dog or something like that. So just a little tidbit for you. So one thing too is sometimes we go a little bit further, and we might divide things a little bit more, and we might have things that we call a subspecies. So the domestic dog, we actually have a subspecies name on there, familiaris, which means friendly. And so, because there are differences between domestic dogs and wolves, although they can breed together, which does make them the same species. So I do want to look at one more organism that is completely different. I'm going to look at one bacterial organism. So this is Staphylococcus epidermidis. So there's a picture of it. And so right from the very beginning, it's different. This is a bacteria. So there's no nucleus, it's a prokaryote. And you can see I made the note about the phospholipids being different from the archaea. It's in the kingdom called the gram-positive. So we're going to talk about what that means when we talk about cell walls. But it has a thick cell wall. It's in something called the firmicutes, which at this point, you know, when we're starting to get to microorganisms, it gets pretty complicated because it's not like they have hair or certain teeth. So you, in this case here, somebody noticed one day that this group doesn't have as much G or C in their DNA. So apparently that's a characteristic of them. It's in a class called the bacilli and the order called the bacillates. And this is based actually on RNA sequences in their ribosomes. So again, sometimes you got to use something different because they don't have a lot of characteristics at that level. So these are in a group called the Staphylococci. So you can see here what this means. Staph means cluster, and coccus means sphere. So you take a look at these cells here, they are spheres and they're kind of in these clusters. So that's what Staphylococcus means. And you're going to see that word again and again this semester, Staphylococcus. And so it looks like a bunch of grapes. So the genus is actually Staphylococcus as well. I'm not sure what, how we distinguish between. Maybe it's just maybe it's a limited group. And then the species is called Epidermidis. And I think I spelt that. Put an extra ID on the bottom there. And epidermis, of course, means skin. And this lives on your skin. So right now, all of us, every single one of us, every human has this on his or her skin. It lives there naturally. It doesn't usually cause us any issues. It's a natural inhabitant of your skin. So a couple other things to consider here. So I kind of alluded to it once or twice here about what is a species. A species from a kind of a traditional biological definition means that if two things can breed, they're the same species, right? So as I mentioned, dogs and wolves, they can breed, they're the same species. You know, some dogs, maybe they have a hard time, you know, there's big dogs and little dogs, but in theory, they can breed so that they are the same species, right? And so that's usually how we define it, but it's not always so simple. Sometimes things can breed, right? So you may know what the answer is here. What happens if you get a donkey and a horse? You get a mule. So it turns out that a mule is not technically biologically a species. Mules cannot breed further. So sometimes you can get these weird hybrids between two different species. And so biologically, biologists would say a mule is not a species and a donkey and a horse is. I'll show you another example here. I found a whole website talking about these. It was pretty interesting. So what happens if you get a zebra and a horse? And so, you know, people are probably thinking is, okay, it's either a hebra or a zorse, right? Turns out the answer is zorse. I'm not sure what's going on here. I'm guessing we've got some people at zoos and farms that are kind of bored and they're making these things, but it turns out a zorse is also not a species. It is a living organism, yes, but as a species, it cannot breed. So therefore, by definition, it's not a species. On a totally related note, I saw a zonkey this summer. Well, that's a zebra-donkey cross. So I thought that was interesting. When we start to get to out of the animal and plant kingdom, it gets really complicated. There's all sorts of fungi and microorganisms that they don't mate, right? There's not like there's a male E. coli and a female E. coli and they get together and have a little E. coli family. It's nothing like that. A lot of them don't mate. Sometimes they swap DNA just because that's what they do, and they don't even have to be related species. And viruses don't reproduce on their own. So sometimes when we get to microorganisms, it gets a little bit more complicated. And you know, we do classify things in the traditional way, which is by characteristics. So we might look at the shape of the organism. Does it have any particular organelles? What can it eat? Can it eat lactose or glucose or whatever? And a lot of microorganisms are also classified by phylogeny, and that's the DNA sequences. And that's a really, really common way to do things nowadays. So just a warning on that. Like I said, you know, when we get to cells, sometimes things can get a little bit more complicated. So I should have put this slide earlier. I wanted to mention how phylogeny is done. Like I said, phylogeny is how related things are, and in today's standards, usually it means, you know, we're looking at their genome sequences. And so with computers nowadays, this is very easy to do. If you have the sequence, there's all sorts of algorithms that puts them together. You can see in my first example, organism 1 and 2 are closely related. In that case, they're about 80% related. Organism 1 and 3, this should say 3, are more distant related. There's fewer matches. And so, if we're 99.9% similar to somebody, we're probably the same species. Humans and chimpanzees, I think it's about 98. And so, that's kind of close, but we're probably not the same species because we can't breed together. But when you look at microorganisms, sometimes it gets kind of murky. So this is one measure of seeing how similar things are anyway. All right, so back to our picture here of the eukaryotes, the archaea and the bacteria. And you can see there's the phylogenetic tree of life. I found a more kind of recent publication showing the tree of life. I think that previous one I showed you isn't even in our textbook anymore. The one they have in the textbook is really ugly. But I found this one, and I thought this was interesting because they actually place the eukaryotes and the archaea a lot closer than they do the bacteria. But anyway, interesting. This is something that biologists love to debate and talk about or reclassify over the years. And a little bit of it's interesting. I don't find it terribly interesting. But what I did want to talk about that's important for Biology 107, for Biology 107, you should know the domain of something. So there were 3 domains, remember? We had the bacteria, the archaea, and the eukaryotes. And you should know the genus and species. Everything else in the middle I don't really care about. What's really, really important is for genus and species that you know how to name things. So take a look at this, right? We have a genus and species, and that makes up the scientific name of something. We call this the binomial name. So we call this the binomial nomenclature system. This is going to be in italics, and so make sure you have it in italics. In all of your lab reports and everything you do for this class, make sure all your species names are in italics. The genus starts with a capital, and the species name is not capitalized. So I want to show you some names and some examples of some descriptive names that fall into this binomial nomenclature system. Traditionally, these names would be done, it'd be usually using Latin. The idea behind Latin is that it's not used by anybody, so you're not favoring the English or the French or the Germans or whoever, right? And usually the best names are descriptive. So you can see there's a cat, Felis domesticus, and the whole idea is that we use the word cat in English and use something else in German, something else in Hindi or whatever, right? Microorganisms, I already showed you one, Staphylococcus, right? So Staphylococcus, staph, remember, means cluster, and coccus means sphere. There's another Staphylococcus. But look at this is a different one. This one is aureus. So maybe you remember your periodic table, and your periodic table has this particular element on it. And usually that's one people remember because that one represents gold. And so it turns out that when you put this particular organism on a petri dish, it's kind of a goldish color. So, this lives on your skin too, but the name epidermidis is already taken. So we had to give this one a new name. So the name was given aureus because it's gold. Here's another organism that I thought this one was cool. I saw this in the Guinness Book of World Records, and you can see it's a coccus, and it has the word dino in front of it. That's the same dino for dinosaur. And if you know what dinosaur stands for, dinosaur actually means terrible lizard. So this is a terrible coccus, and radiodurans means it can survive radiation. So this particular organism was found in a nuclear power plant, and in a place where they didn't think anything could possibly survive. And it's apparently they've tested it, and it's a super tough organism. So that one's kind of interesting. Sometimes things are named after people. So if you take a look, this one here, Escherichia coli, was named after a guy named Theodore Escherich, and he was looking at diarrhea in babies. And so I don't know if he's very happy that they named a fecal bacterium after him, but he didn't have a choice. They did it after he died. This one here, Attenborough, named after David Attenborough. famous naturalist. What about this one? Anyone recognize that name in there? That's right, this one was named after Beyonce. So somebody likes Beyonce or was inspired by her. In fact, if you take a look at this fly, it sort of has a golden color that reminds you of her hair. So, you know, why not, right? There's lots of fly names out there. This one, you know, name it after Beyonce, why not? How about this one here? Neil Pulpa, Donald Trumpi. I know, whether you love him or hate him, you can't deny the resemblance. You know, it's some sort of moth, I think, and it's got a bit of a comb over. So I'm almost done for the day. Just hold on for 30 more seconds. I'll show you a couple more examples of some names. There's another one. Sometimes there's other things there. You can see this one's named after Long Beach, California. Again, I don't know the story behind that one. And sometimes organizations, Legionella is named after, it was discovered at a Legionnaires convention. A Legionnaire is somebody who used to serve in the military, and they got sick from a new bacterium. So just to stress what I said a minute ago, in all of your lab reports, you're going to use your genus and species for every organism that you can, and you can spell it out in full, or you can abbreviate it. Usually the convention is the first time you spell it out in full, and then all the other times you use the abbreviation. You're going to italicize it. If you're handwriting it, you can underline it instead of italicizing it. So keep that in mind when you do all your reports in this class. You must do this or you will lose marks. Always italicize species names. So anyway, we're out of time for today. So I will let you go. Sorry for going over a couple of minutes. We'll pick this up next day right where we left off. Have a great afternoon. We'll see you on Wednesday.